All right, let's go ahead and start. I'd like to, of course, thank the organizers for giving me this chance to speak, and also to the many volunteers who worked a lot behind the scenes, making this actually one of the most interesting meetings that I've been to. So thank you all. And I should also, just in the interest of full disclosure, mention that there's a company that is trying to commercialize some of what I'll talk about, but they were not involved in the research. So I asked Eleazar before whether I should talk about cancer or PopGen, because it was a PopGen meeting, but he said talk about cancer. So there we are. Okay. So let's start with structural variations. I think many of you might be quite familiar with it, but since we didn't talk about it this week, I think it's good to have a few slides talking about how structural variants are discovered. And when I say structural variants, I really mean variants that are 100 base pairs to maybe millions of base pairs in length, and the variation occurs due to deletion, inversion, or some kind of rearrangement or copy number change of these large segments in the genome. And oftentimes, because we're not at the stage where we can fully assemble human genomes in a cost-effective manner, we do this by so-called paired end mapping techniques, where you take your donor sample and you sequence it. Oftentimes the sequencing is done by sequencing two ends of an insert, and the insert can be 300, 500 base pairs, sometimes longer. And then you map these two inserts back to a reference genome, right? So just a few sort of mnemonics, because we'll use these pictures all through and people should be familiar with what they imply. So let's say this is our donor genome, the person we are sampling from, and we've sequenced these ends of this insert, and we go back and map these to the reference. So you use your favorite mapping tool, bowtie, BWA, whatever, and you map it, and they don't map concordantly. So they are 300 base pairs apart here, but here there are 10,000 base pairs apart. So can anybody say what the answer might be? Why is this mapping this way? We could imagine that there's a deletion in the donor relative to the reference, and so if you were to delete this region out, then these two things come close together. So it's kind of a signature of deletion, right? You could also have a situation where they not only map far apart, but one of the reads changes orientation. And in that case, you might imagine that there's a large segment, and if you were to invert it, then you would get a signal which would be like the donor. So it's a signal of inversion, right? And so when next-gen sequencing started in earnest in 2004, 2005, we were studying these kinds of insertions. By the way, there are other signatures as well. Some of them are just by counting the number of reads that map to a location. If you're sequencing a genome to, say, 30 times redundancy, you would expect about 30 reads per base pair. But if you see, say, 300 reads, you know that there's been some structural variation that resulted in increase in copy number. Right? So around this time in 2007, there's this very interesting paper from Michael Stratton's group, who's one of the preeminent sort of cancer genomics people. And they showed this data, and they said the architecture remarkably recapitulates the classic breakage fusion bridge cycles. And for somebody like me who doesn't know what breakage fusion bridge is and can make no sense of this kind of data, there was sort of an eye-opening that people could look at this complex data and actually say something meaningful about it. So we decided to study this in detail. And then over the years, many such examples have been drawn. I'm going to pick a few more. This is Chromothripsis, another very fascinating example where they take a cancer genome and find that the copy numbers are not only changing, but they're changing in this peculiar oscillating fashion. So low, high, low, high, low, high, and so on, right? So you have an oscillation of copy numbers, an extensive rearrangement. And the mechanism they came up with was just as extraordinary as this example itself. They said the chromosome itself shattered into many pieces and then came back together, duplicated, and that's chromothripsis, some kind of a shattering. So again, something quite, I would say, for a computational scientist, it sounds like a very extreme explanation for a phenomenon, and it was something worth investigating, right? But then if you go back in sort of past history, there have been other models proposed. So this is a picture from Jeff Wall from the 70s or 80s maybe, where they talk about small pieces of chromosome pieces of DNA coming out from the chromosomes. They call these various names, episomes, double minutes. We can go a little bit into the history of why these names were there, but not important, but you can imagine that if you have these structures, they may also allow a way of increasing or decreasing copy numbers, and we'll talk a lot about that also. And then finally, we have these classic rearrangement models that we sometimes study in our genetics classes, where a segment, because of some kind of non-homologous recombination, repeat mediated or otherwise, allows you to increase or decrease copies of these large segments, right? So in a sense, this talk is about cancer, but it's also about ascribing mechanisms. You know, we have this incomplete picture where we sequence short reads of whole genomes, map them to the reference, and then we get these peculiar marks, and we should be able to say whether this is Chromothripsis or breakage fusion bridge or episome formation or what have you. Okay, and that's part of where I'm going to talking about today. All right, so let's talk about breakage fusion bridge. And Emilia, of course, when she talked about women not being recognized for their work, gave some great examples. I think Barbara McClintock is another such example. Of course, she was recognized in her lifetime. She was the president of the Genetics Society and also got a Nobel Prize. But I can say in all fairness, her work was not properly understood. Not only that, she never held a tenure-track position in her life. She spent most of her life at Cold Spring Harbor, and she was very happy there. She had good colleagues and so on. She also spent some time at the University of Missouri, where she was, again, on the faculty, though not in a meaningful way. She was just left free to do her own research. And this is where she came up with this beautiful model of a breakage fusion bridge, right? So how does it work? And by the way, she worked on maize, so as a model organism, but the mechanism does have strong implications in cancer because the loss of a telomere is very destabilizing to a chromosome, and I'll show you why it can lead to proliferation and so on, right? So it starts, of course, with the loss of a telomeric element, and because it makes the chromosome unstable, oftentimes when the sister chromatid is created, there's a bridge formed that sort of stabilizes this, okay? So it's temporarily stable, but then when the cell divides and the centromeres move apart, what's going to happen is that these two pieces are going to pull each other and there's going to be a break, right? And the break doesn't happen to be where it happened before, which would mean that a portion of the chromosome duplicated and inverted. That's what you're left with, plus you're left with a broken end. And so this could go on for a few cycles. So if you imagine this going on for a few cycles, in a few cycles, you're going to have extensive rearrangements and duplications of the chromosome, right? And of course, if that part of your chromosome contains a proliferative element or some kind of an oncogene, and it gives the cell some advantage, that cell is going to magnify and could result in a tumor formation. So that's sort of the loose connection to cancer. But the question for us, of course, is whether we can take the paired end signatures from this and predict whether or not breakage fusion bridge happened. Right, so I'm going to skip this slide in the interest of time. It has to do with heterogeneity and just sort of go to the pure problem, right? So the pure problem, of course, is that you take one of these tumor genomes and you take short reads from them, sequence them, map them back to the reference and get counts. And so, this is often what a copy number pipeline would do. It would break off segments and say, this segment A, which by the way could be hundreds of thousands of base pairs long, so we are not talking about small segments, comes with copy number 4, B comes with copy number 4, C with 6, and so on, right? So you have these copy numbers, and you ask if this genome could have this kind of copy number pattern could have been created with breakage fusion bridge, right? So it's actually a very interesting combinatorial problem if you forget the noise that's introduced due to other rearrangements and heterogeneity and so on. And oftentimes I give this in class, for example, all even numbers must precede all odd numbers. And sometimes I give that as a homework if I'm feeling particularly nasty, right? So in this particular case, if you switch the number of A by 1, then it becomes not a support for breakage fusion bridge, but otherwise it does. All right, so let's talk about an algorithm, a sort of combinatorial algorithm for detecting if the copy numbers are consistent with breakage fusion bridge. And the algorithm that we actually came up with, I had a really brilliant postdoc, his name was Shai Zakov. He sort of left academia, unfortunately, afterwards, but he came up with this algorithm. It's very hard to explain. I'm not going to go through that algorithm. I'll talk about a more suboptimal algorithm, which is sort of exploratory, but it gives a little bit more insight into the structure of the problem, right? So let's start. This is not an empty slide. You can see that on the right side, we have this segment, and let's do a few breakage fusion bridge operations, yeah? you can, so the question, of course, there's not a unique scenario that will lead to that copy number, but the question is simply, could breakage fusion bridge explain the bulk of these copy number changes? That's how we sort of give the question. And when we want to actually do it, of course, statistically, we do it against a background of other kinds of rearrangements and say that this one's much more likely to explain, and there are false positives associated with that. So I'm only talking about the really pure case, and even when you have breakage fusion bridge, you don't necessarily have a unique reconstruction, but you have a plausible reconstruction, right? So the goal is not to reconstruct, but just to say yes or no. Okay, so here let's do a breakage fusion bridge, which means we take a prefix, duplicate and invert it, and the only thing I do differently is that I write A on top of A, B on top of B, and so on, right? So we do that for a couple of cycles. And then in the end, we take the entire chromosome and duplicate or invert. And we can always do that by just doubling all the copy counts. And so the input really is these numbers. That's all we have. 12 counts for A, 10 counts for B, and 2 counts for C. And the question is it consistent with the breakage fusion bridge, right? So what you can do is you can try and glue adjacent occurrences of the same letter. So I glued the 2 C's together, and then the B's, and then the A's. And now you can see there's some kind of hierarchy which can be expressed as a tree, right? So you have C out here corresponding to this node out here. It has 5 children corresponding to the B's and C's, and we call these sort of breakage fusion bridge trees, right? The good thing about breakage fusion bridge trees is that they have a lot of interesting properties. They have long ends, which means that the interior leaves are not going to be of height higher than the exterior trees. They have many symmetries, which means that if you take some subtree and you rotate it, you'll get an identical tree. There's also a paired symmetry, which I'm going to skip in the interests of time. And furthermore, if you do some kind of an in-order traversal on this tree, you'll get a plausible breakage fusion bridge sequence. All right, so because you have all these properties, you can start with the numbers and you can sort of do a kind of, I would say, a sort of heuristic search which maintains all of these symmetries, and that constrains the number of possible trees that you can build with this data. And if you can go level by level and end up with a BFP tree, then you have a yes answer, otherwise you have a no answer. So that's not, by the way, that is not the algorithm. The actual algorithm is just way more complicated, and even I cannot explain it in a few slides. It's just impossible to do it. But there's a nice paper, and I can point you to it. So in general, I'll say that the algorithm which we came up with quite a few years back now does very well on both simulated data and in the presence of noise that you introduce. You can see that we are at the point where if you have sufficiently long BFB cycles, then we have about 90% sensitivity given about 10% false positive rate, and so on, right? And we did find a few examples, both recapitulating a handful of previously known examples of breakage fusion bridge, as well as some of our own discoveries. But in the 6 or 7 years since, we have looked and we have found rarely any examples. Unfortunately, it's like a hammer without a real nail to hit it with. So BFB does occur. It does occur. There actually have been a few other reports where they used our tool and detected BFB, but it doesn't seem to be a prime driving force in cancer. So sort of back to the drawing board and say, why are all these complex rearrangements happening in cancer? All right? So I'll give you a negative story next, and this is about Chromothripsis. So the way they discovered Chromothripsis is they said that there's this peculiarity where if you have, this is sort of a cartoon that better explains this picture. You have oscillating copy numbers, low, high, low, high, low, high, and extensive rearrangement. You cannot get that through a progressive model. And the way they did this, they just took a genome and started simulating some kinds of rearrangements in progression. So you start with the tandem duplication, then you do an inversion, you do another tandem duplication, you sort of go along and you accumulate rearrangements on this. And as you do that, you look at how the copy numbers are changing and how these arcs represent rearrangements, how the rearrangements are happening. So obviously, as you do these operations, you'll get more and more rearrangements, right? And you also get many different copy number states. So here there are only 2 copy number states, low and high. But now at this end, because of how the rearrangements happen, you have some with copy number normal, some with 2x, 3x, and so on. So what they propose is that if rearrangements were to happen progressively, and this is from the Stevens model, you would also see an increase in the copy number states, right? So if you plot these two together, on the x-axis, you plot the number of breakpoints of your arrangement, so sort of how many rearrangements you're seeing, and on the y-axis, the number of different copy number states, you're going to get a sort of increasing curve like this. And they found examples that lay completely outside the normal boundaries of this curve and said, hence chromothripsis, right? So now, in a way, if you're a computational scientist, and I, sort of in full disclosure, my PhD in '94, when there was no data, was on genome rearrangements. I worked on these kinds of problems a lot, actually. And this was done only using permutations, no actual data. So, but it's very instructive. But we used some of those tools, and we wrote this paper sort of tongue-in-cheek as 3 strikes against the model. So there were 3 strikes, but I'm only going to tell you one in the interest of time. And this one's simple. You just take a normal genome and you do what we call copy-neutral rearrangement. So you do a lot of inversions, right? So you take a segment, 2 boundaries, and you just invert it. When you invert it, everything changes and their signs also change. And if you do a few inversions, you'll see a huge number of rearrangements, which is pretty obvious, right? So when you map all these back, you will get the B to A coming out as an edge here, the C to B going like that, and so on. So you will see a graph with many, many edges, but absolutely no change in copy number. You do that, you do a single deletion, which removes a huge chunk, and then you do maybe 1 duplication, and if you map it back, you will get what they called was the signature of chromothripsis, right? So it's a really extreme and stupid example, but if you actually do these examples and you plot them, this is where our simulated chromosome lies, right next to where they claim chromothripsis happened, right? So the point is not to say that chromothripsis occurs or doesn't occur, but that the statistical evidence is somewhat weak. And of course, when you write a paper like this, it's impossible to get it published, and it took many years and sort of sympathetic edit to get it published, and we did publish it. But it's interesting to actually think a little bit about the critique of our approach. So there were many, of course, we really had, it took some time for us to publish. One of them was that there was some experimental evidence for chromothripsis. In particular, Pellman, they did some really fantastic experiments where they sort of made these movies of images of cells, which due to some kind of phenomena, the chromosome lags during cell division. And once it lags, it sort of, it just gets shattered apart and shunted into micronuclei, and that's one mechanism in which chromothripsis can happen. So first, we would like to say that we did not claim that chromothripsis didn't occur. It's just that the signatures that we have, which are based on paired end sequencing, which everybody uses, may or may not be sufficient to detect chromothripsis. The other sort of other criticism was even more instructive. It said, what you're doing is completely unrealistic, right? You don't have genomes where there's a lot of inversions followed by some deletion, duplication. Even if that can explain the chromothripsis signature, it's not a realistic model. So you're only poking holes, you're not really giving an alternative, right? So today I'd like to present a sort of semi-alternative, right? And this is interesting work. It came out of a thought that we had, which, if you take sort of the classic model of episome or double minute formation, what happens out there is that you have these chromosomes and you have little segments that break away and circularize and form sort of these molecules that are outside of the chromosome, in the nucleus, but outside of the chromosome. And these can actually replicate, and so they make many copies. Now, if you take your bulk cells which have these episomes, you sequence all the DNA, you map it back along with paired end mappings and so on, well, what's going to happen? You're going to get these oscillating copy numbers because these are the only segments that have increased in copy number, and you'll have the rearrangements because they come from different regions of the chromosome or even multiple chromosomes, right? So in a way, this very simple model, which was discovered way back in the 80s, can somewhat explain oscillating copy number patterns and complex rearrangements, right? So this is where we were when I started a collaboration with the Paul Michel's lab. They had come out with a really fantastic experiment that I'm actually going to tell you about in a few slides. And after talking to them, we did a pretty large experiment. We took about 143 samples spanning many different tumor types. So you can see we have all these different tumor types here, tissue of the tumors, and we did what's called a metaphase fish experiment. And for those of you who are not familiar with that, I'll show you a picture. Basically, you capture the cells at a time when the chromosomes are compacted. And because the chromosomes are compacted, you can sort of see if there's extrachromosomal elements, they will show up. And if you stain all the DNA, the chromosomes will show up as sort of compact chromosome, those X shapes that you often see, and then the ecDNA will, the extrachromosomal elements will show out as little dots, okay? And so we took about 2,000 such images, and now we could not look at them by eye and look at, what was happening to ecDNA, so we built a little image analysis tool which takes an image like this. So this is a metaphase image. These are intact nuclei, for those of you who haven't seen it. These blobs are chromosomes, and then out here are these little dots are what you would call extrachromosomal DNA. And the algorithm itself is somewhat boring for those of you who've done some image analysis, but basically it starts by identifying the nuclei and then the region of interest, and removes the nuclei because those are not interesting. Within the region of interest, it first tries to identify chromosomes, just using very crude criteria, connectivity, size, so on. And then within that, it tries to identify extrachromosomal DNA, and then it returns a count. Okay, so that's what ECDetect does, and it works fine. We have very high specificity, and this is, by the way, this is an older version of the tool. We have about 99% specificity. So when we predict something as being ecDNA, we know that it is ecDNA, but we only have had about 50% sensitivity, and that had to do with the fact that many of these extrachromosomal elements were very, very close to chromosomes, and we couldn't really distinguish them from the boundaries. And since then, like everybody else, we have sort of gone to a deep learning approach and have better results, but I won't talk about them today. All right. So this is what we had at that time, this tool, and when we applied it to our images, I think we were shocked. Okay. So the thing you have to remember is that there's just way more ecDNA than you would have thought. We found that 50% of all the samples that we looked at, right? So 50% of all samples and every single tissue type had an abundance of extrachromosomal DNA. And just to say how startling this estimate was, the previous estimate was only about 1.5% or 2%. This came out of a paper actually from David Hausler's group, where they were looking only at sequence data. And there's a lot of, I'm going to explain why sequence data for all its power has some weaknesses which are not completely apparent, and those can cause problems in interpretation. So there weren't many great estimates, but our estimates suggest that these are ubiquitous, and it really should change our thinking about cancer. Yeah? They're not, so we don't see them in normal. Okay, we rarely see them in normal, although I will say this. Okay, so since you ask, I want to just point out that we do see, there's a paper from Anindya Bagchi's group in Virginia and from Andrew Fire's group. They've identified small extrachromosomal circular DNA in normal cells, but these are only 100 base pairs. And the reason they only identify these is the technique is completely different for identifying based on some trap. They cannot identify longer ones. There's some suspicion that they happen, and then they enlarge through recombination, and then once in a while they proliferate. And I'll talk a little bit. I mean, my talk is about this model, so I'll come back to it. Okay, so just to say, these are sort of these violin plots, but on a log scale. So some of them, like glioblastomas, are just completely abounding in these, and there are many other cancers where there's a large number, and then there are a few where they're not quite as large. So there is some tumor subtype specificity to this, but by and large we see them. The other interesting thing is that when you look at patient-derived xenografts in general, you see many more than you see in tumor cell line. That's because the metabolism of a patient-derived xenograft, which is like a three-dimensional sort of tumor, which is what would happen in the body, is very different from a flat surface where there's a lot of sort of metabolites available. And the role of metabolism in controlling the number of ecDNA is also quite profound, something that we are working on, but I won't report on today. But I just want you to know that there are a lot of differences, which is why when you study cell lines, you may or may not see them as often as if you were to study, say, xenografts. All right? So one of the questions that people often ask us is, how do you even know that they are circular? And we actually didn't have an answer when we published our paper last year. But since then, my colleagues have done some electron microscopy, and you can see that there are, and these are really large. You have to say, if you also want to know how large they are, these ones are close to 10 million base pairs. Okay? And we routinely see them between 1 and 5 million base pairs. All right. So let's talk about, I don't have a lot of time, but let's talk about the main topic that I wanted to argue today. It's an ecDNA derived model of copy number amplification in cancer. right? And so the way this model works, the proposal, so fragments break off at random and form extrachromosomal elements. So you see that here, right? Now these fragments have origins of replication are quite common on the human chromosome. So it's quite likely that they have origin of replication which can fire, and these can make copies independently of the chromosome replication, right? And recombination can lead to larger copies, multiple copies, and so on. Now, when the cell segregates, these are not bound to the centromere, right? So the centromeres pull the chromosomes so that an equal number lie up in daughter cells. ecDNA do not have this restriction, so you could have an ecDNA here which made 2 copies and both copies ended up in a daughter chromosome, right? So that's number 3. These are not bound to chromatin in the same way that normal chromosomes are, and so they could be completely dysregulated, something that we are studying quite extensively now. And if they are completely dysregulated, it could be that they are overexpressing a lot of stuff that's on there. And if you get unlucky and there's some kind of an oncogene, you will get a proliferative advantage because this gene is making many, many copies of the protein. It's leading to growth signals. And so if these cells make many more copies than the ones with fewer copies, then you're eventually going to start seeing an increase in copy number. And you know, we did a lot of simulations in our Nature paper, and of course we show that, you know, if you have a tandem duplication model versus an ecDNA model, you can rapidly increase in copy number. And the most pernicious cancers, the one that sort of don't progress over many years but just spring up and sort of kill you before you even know where you are, they could be derived from these kinds of extrachromosomal DNA elements. All right, so you have this, and then the final and sort of more controversial part is that they sometimes reintegrate back into the chromosome at non-native locations and show up just like a tandem duplication might show up, giving a completely misleading picture of where they actually came from. Okay, so this is where we are. And if you take all these cells and you collect them in bulk like we do and do whole genome sequencing, a lot of you get very precise breakpoint estimates and so on, but a lot of information goes missing. Specifically, the heterogeneity of these cells is not well understood. Where these cells are completely misunderstood. So you have many examples of people doing association analyses with copy number changes, but think if these copies are coming from different portions of the chromosome, what value can that association analysis have? So there's a lot to think about. People also do analyses of somatic variation and their evolution. Sometimes people think about their allele frequency spectrum, but if the evolutionary model for ecDNA is different than the somatic mutation, you don't want to put them in the same model, right? So for the young people in the audience, I think it's definitely worth thinking about these things as you're working on some of these problems. All right, so here's our model, of course, and how much time do I have, by the way? 15. I have some time. Okay. So in the time that I have, I'm going to talk about proving each of these aspects of this model. And proving, we had a very interesting conversation with Siavash and Ilana yesterday. We were thinking about proofs, and I'm going to say this in jest. So for the experimentalists here, please excuse us, but there are many computational scientists, so I feel safer saying this. So, let's say your goal is to prove some kind of an assertion P, right? And we know that P implies Q, that if P is true, then Q is true. Oftentimes in biology, I don't know, this is funny, but we make this thing that if you see Q, then P is true probably. But actually, biologists are a lot smarter than that. They don't say that this is a proof of say, but you say that the observation of Q is consistent with the hypothesis of P. And it's actually a good way of thinking about things. Something, even though there's a clear logical fallacy here, it's really a smart way to do these things. The way you can do it, just to strengthen your argument, you don't have to do that P implies Q, but you might have P implies Q1 and P implies Q2 and P implies Q3. And if you see Q1 and Q2 and Q3, I think you can start believing that P must be true, because the knot of that begins to get reduced, more and more reduced. And this is a good way because you can sort of experimentally do things. So it's in this spirit that I'm going to prove different tenets of the model. And so if you see the logical fallacy, don't say I didn't warn you. Okay? And of course, I'm also not going to do them in order because I don't want to waste your time with different things. So let's talk about this one. Okay, lack of centromere implies random segregation in daughter cells. It's sort of number 3 of our model. And of course, what this implies is that if you were to look at cells individually, you should see a lot of heterogeneity, right? In one cell, you would see some counts. In the other cell, you'd see another counts. And it's clear that we do see them. We can do this with standard sort of entropy measures. So each column here represents one sample, but many cells taken from that sample, and then we use our software to count, and we just compute the Shannon entropy. So it's very simple. And you can see that there are some cells which, normal cells, have very little heterogeneity, but if you look at brain tumors and patient-derived xenografts, it's tremendous heterogeneity. It's as bad as you can see. And here are sort of more concrete examples. This is the same. This is a cancer cell line. I'm sorry it got erased out here, but you see no ecDNA here, a few here, and many here. And you can see that there's heterogeneity, right? So that's our proof that that there is there's random segregation of cells. Okay? So presence of oncogenes provides selective evidence advantage and accumulation of ecDNA, right? So what we did was we took all the TCGA data, but we were lazy. We didn't take the whole genome sequencing data. We took array CGH data because we were only looking at copy numbers. We have since gone and built structures. I'll talk a little bit about that maybe, on all TCGA whole genome samples, which was like 20,000 hours on the GPU cloud, and at some point I'll report on that. But for now, we just took array CGH data and we saw the fraction of samples in which we see an amplification of an oncogene, right? So EGFR is a good example. You see a lot of them. On the right side, we took the 140 samples that we had and we counted the number of times you would see a particular gene on an ecDNA, and you can see that there's a very tight correlation. We have a statistic too, but just visually you can see that there's a very nice correlation. The ones that we see are ecDNA seem to carry the same genes that are known oncogenes and are amplified in the broader TCGA sample. So for those of you who are not aware of TCGA, it's a large sort of pan-cancer project where they took many, many samples, thousands of them. These are not cell lines, these are patient samples, and they acquired a lot of data on them. The array CGH is somewhat older now. They have exome and whole genome and transcriptome and all these data sets, and so there's a lot more data available. Here's sort of the most fascinating part of this model. ecDNA may integrate back into the chromosomal region and thereby explain some focal amplifications. And actually the genesis of this was a paper written again by my colleague Paul Michel many years, some years ago, and it really began, it was the start of our collaboration, to be honest. They did this really beautiful experiment where they took a glioblastoma cell line, it's a brain cancer, it's very sort of deadly. You wouldn't want to have it, and it's characterized by an overexpression of the epidermal growth factor receptor. Okay, and so what they do is they again take a metaphase spread of the cell. The blue color is staining for all the DNA, so you can see all these are chromosomes, and then they have a red marker for EGFR, and you can see that the red there's a lot of red dots here. I don't know how visible it is. Yeah, it is visible, but they are all outside of the chromosome, suggesting that these EGFR elements are on extrachromosomal elements. And then they feed this with a drug, erlotinib. It's a blocker for the EGF receptor, and some cells die, of course, because of that, because EGFR is involved in glucose transport, and glucose acts as a substrate for extra DNA, and you need DNA to make these extrachromosomal elements. And when you give this drug, all the extrachromosomal elements disappear, and they all seem to, the light all shines on a chromosomal region where they act like homogeneous staining regions. So this is an archaic term, but you can just think of it as being intrachromosomal. It's a region of the chromosome that's sort of stained homogeneously, so HSR. And so what has happened, what one might suspect has happened, is that these extrachromosomal elements, some of them have integrated into a chromosomal location. And then you can do an experiment where you also light up the centromere of a specific chromosome, and that way you can tell if it is non-native or native in its location, all right? And then when you remove the drug, these elements come back out again, right? So of course that leads to many questions. We have no idea if it's the same element that's coming out. We know it contains EGFR, but does it have the same structure? So we go back to the drawing board, and my student Viraj Deshpande, who's since graduated, built this tool called Amplicon Architect. And the goal for Amplicon Architect is very simple. It reconstructs the fine structure of the extrachromosomal DNA using this paired end sequencing data. So it's not like a de novo assembly algorithm, but it starts with the mapping of these reads to a reference, and it builds off of those references. All right, so you start with these mappings, and you can see that there's some expansion and you give a seed interval because you are interested in EGFR. You can say, I want to build an amplicon around EGFR. And the way it works is it first takes this region and looks at where you have a lot of support for a discordant reach, because there's a lot of noise in the data, but you only look at the discordant reach for which there's a lot of support, and that allows you to chain this region with another and sort of get a larger region. Once you have a larger region, you do some kind of a smoothing to get curves. It's very similar to how people do copy number analyses, but we sort of smooth out, and we can see at this point that this region has an identical copy number, but then there's a sudden jump to another copy number. And the sudden jump simply indicates that this segment has made many copies. Those copies don't have to be co-located, but there are multiple copies. And so at that point, it looks for additional breakpoints that explain these copies. So these are reads that might say that this segment is now connected here. And then finally, when it has sort of a region of the genome that it's interested in, it goes into the noise and picks out a few more copies. And that's what gives you a sort of amplicon graph. And if you draw cycles from this graph, they're not identical to the extrachromosomal cycles, but they could be representative, right? So and the picture looks something like this. You have this graph out here, and here are some of the cycles that you can pull out. And the way we pull out these cycles is we run some kind of a combinatorial flow algorithm that smooths out the curves. And you want to pull out a cycle, so because cycles should have all identical copies. So every segment you should pull out an enough number of copies, and you do that till you've explained 80% of the copy number increase in the genome. And so you get a bunch of cycles, and the hope is that they cover, they have some kind of a basis representation of all the cycles, and any cycle that might really exist, you can get by recombining some of these cycles. And in fact, there's a lot of dynamism. You don't see exactly the same kind of structures everywhere. So I think this is a good way to approach. And of course, our paper on this is still struggling in revision, but hopefully it'll see the light of the day soon. So let's go back to this example where we had this EGFR. You put the drug in, it became an HSR, and then you remove the drug and it came out. When you run these different samples independently through amplicon architect, so we have 3 replicates in the untreated case, there's one in the drug treated case, this is the drug removed, and this is another untreated sample. And the EC and the HSR classification comes from the cytogenetic experiment. So we know that these are, and you can see that the structure is nicely preserved in the replicates. When it goes down to HSR, there is still a little bump, but there's a decrease in copy number. And furthermore, you see an increase in another copy number which didn't come out before. And then when you remove the drug, you see a more complex structure that incorporates the previous structure almost exactly with a little change, and another MDM2 containing, so a more pernicious extrachromosomal DNA. Okay? And that's not the only example. We have since looked at many such cases where you have things going in and out of the chromosomes and maintaining essential properties of their structure without. All right, so we have good evidence to suggest that these things can move in and out of chromosomes. All right, so let's talk about one more, which is that dysregulation should cause overexpression of oncogenes, and if there are proliferative elements that will provide a selective advantage. So it's sort of intuitive, but it requires some kind of analysis. So we took, and it's sort of an aside story, but we decided to study around the same time cancers that are mediated by viruses. So many of you know that cervical cancer, for example, is a good example, but also head and neck cancers and others. A lot of the times the pathology, not always, but a lot of the times the pathology is explained by a viral genome that inserts into the human genome, and it causes a lot of rearrangement. And I can tell you there are lots of papers where they talk about how these viral genomes induce really complex rearrangements. And we suspect, we didn't have the proof until now, that these could be because there are hybrid extrachromosomal elements created which are part virus and part human. All right, so before we do that, we have to find the place where the virus inserts, and you might think that's an easy problem, but we spent a couple of years trying to solve that because the tools just don't work very well. And the reason is that these viruses, the HPV is only a 7,000 base pair virus, and you have little fragments of it in the human genome. The human genome already has remnants of many other viral elements, and there's a lot of heterogeneity in the sequence itself. So it's actually tricky to find this. And my postdoc, Nam, he built this interesting thing which we call an ensemble of HMMs. it takes the entire phylogeny of HPV, and for different clades of the phylogeny, it builds different HMMs, and then it takes all of those HMMs and searches, and that allows them to find these sort of human insertions in the viruses, okay? And so we find these regions, we get this region, sometimes there's amplification, then we run amplicon architect on it, and we end up with these structures that are very reminiscent of cyclic structures that one might find in extrachromosomal DNA, and are furthermore hybrid. So this part is virus, and this part is human, and the scale is not right. So virus is 7,000 base pairs, the human is 100,000 base pairs, all right? So, but this one we got lucky. It has a gene, which means we can probe for it cytogenetically, all right? So because our paper is in review and the reviewers are demanding, so we also did some PacBio sequencing on this, and we can show that there are reads, PacBio reads, by the way, you know, about 30% of the reads are over 40,000 base pairs, so you can find reads that chain together multiple breakpoints, and you can see that there are these nice reads, and they chain together these viruses, and you can see that this is a very complicated structure because you have the same viral element replicating, but if you combine these two reads, you get very nice cycles that are completely also obtained with the short read data. Of course, the short read data cannot predict the cycles uniquely, but nor can the PacBio data because neither of them is long enough to fully encompass. So we are confident that these structures are more or less correct. Now we did a fish experiment, and you can see that there's actually an extrachromosomal element when you stain for DNA. There are other examples like this. And then if you look at a probe which only highlights the FOXC1 gene, you can see that it lights up, suggesting that there's a human extrachromosomal element containing the FOXC1 gene, suggestive of an extrachromosomal element. All right, so we believe we have the proof for this hybrid. Can I take 2 more minutes, or yeah? All right, so I'm going to try and wind up quickly. When we look at the structures of these and we overlay some transcription data, you see something quite amazing. So we have the viral part here. There's a part chromosome 3, a part of it is chromosome 2, and there's a viral regulatory element. And so not surprisingly, you see a 1,000-fold expression of the viral gene. These are, by the way, known oncogenes, one of the mechanisms. But we also see a lot of fusion mRNA, and we see a tremendous expression of everything in this extrachromosomal element. What is remarkable about this is that there's no gene on this human genome. It's just complete dysregulation. Everything that's on the circle is getting transcribed. And so here's our hypothesis. You know, there are, of course, ubiquitous line elements and LTR, these sort of inactive elements in this. And there's a lot of indication that when these elements are expressed, they can cause a lot of DNA damage. So maybe the mechanism is not oncogene driven, but your transcription drives the expression of these sort of otherwise silent, remnants, fossilized elements, and when they are expressed, they cause the damage. All right. So I think, I'll conclude by saying that we went through a very, very long story. We started looking at complex structural variation, which are found in cancer, but I want to at least, sort of argue that you want to study these separately and not in this crude context of associating copy number changes with phenotypes, because the real answer is a lot more interesting and complex than one might say. There are a lot of mechanisms, but it seems that these simple episomal mechanisms are the dominant ones, and there's a lot of work that one can do to express the biology of these elements. Okay, so I would definitely like to thank Paul Michel. He's a great collaborator of mine. He's just a wonderful scientist, and we talk pretty much every day, but there are also many talented people who work with me. Shai worked on BFB, and Marcus worked on the Chromothripsis project. Viraj did this amplicon architect tool. Doruk Bader built the EC detect tool, and Nam did the human tumor viral integration. All right, so I think I'll stop here. Thank you, guys. Yeah. Yeah, I mean, when we started the project, all the microbiologists who looked at projects said that this could be done because these are like little, bacteria that are inside, but it doesn't work so well. And the reason it doesn't work so well is that the way it works is that you try to entrap these circular elements and you digest all the linear DNA. But there's a lot of linear DNA to digest. These elements are 1 million base pairs, but linear DNA is like 1 billion base pairs. And so what it ends up, we did an experiment where we just undersampled the whole genome, and we did this enrichment, and we sequenced the enrichment, and we couldn't see any difference in the two signatures. And in fact, all of the data that we sequenced and assembled using amplicon, except the one that we did for TCGA, was done with this enrichment. But in a way, it didn't really help us very much. But that's to say, other researchers, including Fire Lab and all, are building these techniques, and they are trying to isolate, and having pure elements would definitely help. Yeah. Okay, thank you.